Good afternoon, everybody. I first like to thank the OIS. My name is Jing-Feng Huang, and I'm the founder and CEO of Seinda Biomedical. Now, just to relax a little bit, because it's been a very intense afternoon. So I like to ask the audience one question. When you see Seinda, our company name, what do you think of it? Last year, just right before ARVO, a friend of mine told me, super efficient IND and NDA. When I picked the name, I didn't think of it that way. Another way to interpret the name would be science, efficacy, NDA, IND, and NDA. So with that, I'd like to tell you what we do. So we have two businesses. One is tier-based biomarker diagnostic. The other is therapeutic. And that's based on very extensive biomarker analysis. Because we believe in order to develop effective treatment, we have to understand the underlying mechanisms of dry eye. And tear film is a great place to start. And what are the molecular mechanisms of dry eye? And I borrow Dr. Steve's diagram here. We know at the ocular surface, breakdown of corneal barrier, damage of the tight junction, loss of goblet cells, and mucin production, and of course, a lot of inflammation. Now, if we look at a given patient at the ocular surface, I think the microenvironment is very important. And how do we assess that? We believe component of the tear film, changes in the tear film component could tell us about it. And that's why we go about biomarker analysis in dry eye. And what I think we often forget, mucosal tissue, which our ocular surface is actually quite different from blood, because our blood system is a sterile system, whereas the mucosal surface is like our intestine. So its immune regulation is actually quite different. Homeostasis is important, even with the immune system. So what about our technology for biomarker test? Because there's so little tear, so we first work on developing a high sensitive and precise diagnostic technology so that it can couple with the biomarkers we identify to develop good point-of-care diagnostic that can be used by the doctors easily and liked by the patients. So the advantage of our technology is, compared with other that's on the market, we have a tear collecting device that collects precise volume of tears and then apply to a highly sensitive and precise test card. And then we use an analyzer to give us a quantitative results. And we use this platform, develop a series of products for different eye conditions because we believe in precision medicine for eye care. And these are some of the products we already developed, and two of them already commercialized in China. And the first one listed here, Lymphotoxin alpha. Based on large amount of data, we know this test can help us separate dry eye patients that are deficient in goblet cells versus the one that with very high level of T cell activation at the ocular surface. Without going through into other tests, I'm going to tell you more about this test. So what is lymphotoxin alpha? It's a cytokine that belongs to the TNF family, and it's very important for mucosal immune homeostasis. And based on the large number of data we have, we know that it's significantly reduced in dry eye patients, in a very high percentage of dry eye patients. And its level is more reduced in the more severe dry eye patient. And we also have a large validation clinical study wherein we enroll more than 1,000 patients, 1,000 subjects in China. And when we take the pre-specified cutoff point of 0.8 nanogram per ml, we find that more than 80% of the dry eye patients have levels below that, which that means our positive agreement is more than 80%. And because the product is launched, so based on real-world data, we see that clinically diagnosed dry eye, 70 to 80% of the dry eye patients in the real world also have lower level of LTA. And furthermore, we also know that the level of LTA in the tear film strongly positively correlated with goblet cell density. This is very interesting, and I believe this is the first discovered phenomenon. And what that tells us is that by testing LTA, we can indirectly assess goblet cell deficiency and mucin deficiency. And currently, there's no easy way to assess that, but we know mucin is extremely important for tear film stability. And we also know that with the 80% of the LTA low dry eye patient, its tear film biomarker profile is different from normal subject. It's also different from LTA high subjects, where it doesn't have that much T cell signal. It's the LTA high subjects that have more T cell signal. So taken together, we have one biomarker test can help stratify patients into two groups and direct treatment selection. The LTA high would be more responsive to cyclosporine, which we have data support that as well. So now I'm going to take the last minute to tell you about our therapeutics. And we are finishing our phase 2 study in the US, and our readout will be early next year. And we have over 100 IIT data. And we see that in 2 weeks of treatment, we have significant improvement in corneal staining. And here's the design of our phase 2. And now I'm going to just want to invite whoever is interested in collaborating or discussing, please, we would love to interact with you. And we are looking for strategic partners for both our therapeutic and diagnostic, and we're also looking for investment for our phase 3. Thank you.